Hello. Today we will be discussing cellular aging and senescence and the role repressor activator protein 1 plays in this process. It has been proposed that there are 9 hallmarks of aging. Here we will focus on 3 specific hallmarks: cellular senescence, telomere attrition, and epigenetic alterations. More specifically, we will look at how senescence can be induced by the shortening of telomeres. An accumulation of senescent cells causes inflammation, which can then lead to many diseases such as progeria, cardiovascular diseases, cancers, metabolic diseases, neurodegenerative diseases, and others that are related to stem cell dysfunction. Understanding the mechanism of senescence and the molecules involved can help us develop therapeutic agents against these diseases. Cellular senescence, or cellular aging, is a biological phenomenon in which somatic cells cease to divide and irreversibly arrest in the G0 phase of cellular replication. Senescence is caused by 4 general factors: epigenetic dysregulation, telomere shortening, DNA damage, and mitochondrial dysfunction. Telomeres naturally form a T-loop structure at the end of chromosomes that is stabilized by a complex of proteins known as the sheltering complex. This complex helps to prevent detrimental DNA damage responses from being activated due to the shortening of telomeres. Somatic cells, which regularly undergo senescence as a natural phenomenon, lack the telomerase enzyme, which elongates telomeres after every replication cycle. Therefore, as these cells begin to divide, their telomeres gradually begin to shorten, leading to the shortening of the T-loop. This causes the sheltering complex to become destabilized. Eventually, telomeres will lose their sheltering complex, thus activating DNA damage responses that induce senescence in these cells. A specific protein involved in the sheltering complex, the repressor activator protein 1, or Rap1, plays an interesting role in what happens after DNA damage responses are induced. Rap1 has been shown in Saccharomyces cerevisiae to help regulate telomere length and transcriptional activation of mRNA encoding genes and telomere proximal genes. It has also been shown that Rap1 relocalizes to new sites on the chromosome at senescence. In this figure, we see a chromatin immunoprecipitation assay using anti-Rap1 antibodies. The plot above shows the chromosomal distribution of Rap1 wild-type cells on chromosome 7, and the plot right below shows chromosomal distribution in senescent cells. We see that Rap1 relocalizes to new sites on the chromosome at senescence. The second half of this figure shows that Rap1 occupancy at telomeres is decreased at senescence. In addition, it was also found that overall Rap1 levels do not change during senescence, as seen in this immunoplot. Next, we will see how Rap1 has been shown to drive senescence. In this figure, we see population doublings plotted against cell density for 4 different types of cells: wild-type cells, cells with decreased Rap1 levels, modeled senescent cells, and modeled senescent cells with decreased Rap1 levels. We see that as these cells increase in population doublings, the wild-type and Rap1 deficient cells remain relatively constant in their cell density. However, in the senescent modeled cells, we see that reducing the levels of Rap1 significantly delays senescence, meaning that Rap1 potentially drives senescence. In addition, histone levels have also been shown to help drive senescence. Here, we see wild-type cells, senescent modeled cells, and senescent modeled cells that overexpress histones. Increasing the levels of histones helps delay senescence. Now, how are histone levels and Rap1 levels linked? As mentioned before, Rap1 protein relocalizes to other sites of the chromosome. Specifically, it has been shown to relocalize to sites upstream of promoter regions of hundreds of new target genes. These new target genes have been shown to be preferentially activated at senescence. A key characteristic of senescence is the loss of core histones. Here, we see an immunoblot of whole cell extracts assaying for histone protein levels. We see that wild-type and pre-senescent cells have comparable levels of histone protein, while senescent cells show a dramatic decrease in the levels of histone protein. Rap1 has been shown to target and suppress genes that encode for core histones. This next figure shows mRNA levels of histones in 4 different types of cells: wild-type cells, cells with decreased levels of Rap1, mottled senescent cells, and mottled senescent cells with decreased levels of Rap1. We see that the repression of core histones is dependent on Rap1, since Rap1 deficient cells have similar levels of histone mRNA compared to wild type. However, the mottled senescent cells that have decreased levels of Rap1 have the lowest levels of histone mRNA. Finally, in this last figure, we see the relative gene expression of histones in cells that overexpress Rap1. We see that increasing the levels of Rap1 blunts histone gene expression. In conclusion, much is still left to uncover about the Rap1 protein, such as the mechanism through which it acts as a transcriptional activator. Also, it is not known how Rap1 represses histone transcription when it relocates to histone promoters. There are many other things that are still left to be elucidated. Answering these questions can lead to major therapeutic treatments in the fight against cancer by suppressing tumorigenesis and learning more about the role it plays in tumor suppression. In addition, it can also lead to treatments in other age and inflammatory-related diseases, such as progeria, obesity, diabetes, and immune system diseases.